Okay, in case you were absent or just want to show your parents what we did or review, I'm going to go over what we did in class this week. One day we did a prokaryote search, and if you'll remember, a prokaryote is a type of cell that doesn't have a nucleus. It's not very advanced, it's just got free-floating DNA. It's usually things like bacteria, usually single cellular types of little critters. So we decided to look for some of those in a sample of Lake Ray Hubbard water. And what we did is first we found something we could see, and we focused on that on our red objective, and then we zoomed in to the yellow objective, and then we just sat and waited, and with any luck we would see some things zooming and zapping around in there. Most of us did get to see some things, some of us didn't, but it was fun to look anyway. Next we looked at an onion cell, and obviously an onion is a plant, and so it's a eukaryote. This is what it looked like on 40, 100, and 400. You could see it looks a lot like a brick wall. The plant cells are very thick. The walls are very thick like this. And on these particular cells, on the onion cells, the nuclei are pretty big. You can see them right here. We also looked at leaf cells, and these also look a lot like bricks with the cell walls being very thick, 40, 100, and 400. Now, because it was a leaf, we could see lots of chloroplasts, the green things that make up chlorophyll, that make the plants green. And we could see so many organelles inside each cell that it was really hard to decide which one was the actual nucleus. And when we compared them, these are both at 400x, when we compared them, we saw that the leaf had much smaller cells, the onion cells were much larger, the nuclei were much larger, but they're both eukaryotes, they're both autotrophs because they're both plants. And then the last thing we looked at were cheek cells. We stained them with iodine. This is at 40, 100, and 400. And what we did is we found one thing that we wanted to focus on. I picked this, and I moved my pointer to point at it, and then I zoomed into 100x. So you can see that's the same structure right there. And then 400x. We've got that in the bottom. And when we got to 400x, it was pretty easy to see the nuclei inside the cells. And the coolest thing about this particular slide is if you look right here, you can see that this cell almost looks like it has two nuclei, one on this end and one on that end. And what's actually happening there is that cell is splitting into two cells. Now, we weren't lucky enough to see that with all of the slides that we saw, but at least I did get a picture of it, and that was pretty exciting. Well, I hope you enjoyed our microscope work today. Up next, we're going to be working on taxonomy, and taxonomy is pretty interesting too. It goes with all the cells and the life and everything you've worked on so far. And have a great day.